All right, so now we are moving on to chapter 11, which is forms of antimicrobial control, and this would be specifically the physical, chemical, and mechanical methods. So chapter 12 will focus on antimicrobial control through the form of antibiotics or antimicrobial drugs. So for chapter 11, these are all of the physical, for example, temperature, chemical, for example, bleach, and mechanical methods, an example would be filtration, to destroy or reduce undesirable microbes in a given area or to decontaminate a specific area. And the targets usually involved with causing infection or spoilage would be the following. So vegetative bacteria, endospores, fungal hyphae and fungal spores, along with yeasts, protozoal trophozoites and cysts. So remember, these are the dormant form of this organism. The helminths, viruses, and prions. So for physical methods, we will go over how changing the temperature, especially increasing the temperature, will allow for a form of antimicrobial control, and usually it works better in the case of moist heat than dry heat, but also reducing the temperature. So if we have a cold environment, this will work a little different than how heat will function to inhibit the growth of microbes, but it still can be effective, specifically serving a microstatic effect, not necessarily microcidal like heat does. We also will talk about different forms of radiation, both ionizing and non-ionizing, different chemical agents that can be used to destroy microbes, so both gases and liquids, and then mechanical, which would be in the form of filtration. So there's a few terms here throughout this chapter that we should all be familiar with. Number one, disinfection. This would be the destruction or removal of mostly vegetative pathogens, but when you disinfect something, you do not necessarily destroy the highly resistant and hardy endospores. And the application here typically is only on inanimate objects. When you sterilize something, this would allow for the removal of all microbes, including endospores. And this would also be used on inanimate objects. Whereas in antisepsis, this would be reducing or inhibiting the amount of vegetative pathogens, but the application here is on a body surface, usually on the surface of skin. Now we're going to talk about the general relative levels of microbial resistance. So as far as the highest resistant associated microbe, this would be prions or prions, and also bacterial endospores. At the bottom of the list, the most sensitive to forms of antimicrobial control, most vegetative bacteria, fungal spores and hyphae, along with yeast, viruses that have an envelope, and the active form of protozoa. The ones that are associated with moderate resistance are Pseudomonas, Mycobacterium, TB, staph bacteria, and also the cyst or the dormant form of protozoa. So this is actually just showing you unique to endospores, the extremes in different methods here to destroy them. So if you check it out here, compared to vegetative forms, so the active forms, for heat, much higher temperature, so they're more resistant, much higher dose of radiation and sterilizing gas, along with using glutaraldehyde that should act to kill the spore. So 3 hours to 10 minutes, so much more resistant here. So we went over these three terms. Also, if you hear the term sanitization, this would be a cleansing technique that will mechanically remove or reduce the number of microbes, and the same with degermation. So this is just reducing the number of microbes through mechanical means. So washing your hands, for example, or washing utensils. Now, as far as decontamination and sepsis, so we mentioned antiseptic. This would be applied to body surfaces. For sepsis, this is the growth of microbes in tissues. Asepsis would refer to techniques that prevent the entry of microbes into sterile tissues. And decontamination, this is referring to the destruction, removal, or reduction in undesirable microbes. Now, as we're going through, and we will mention each physical, chemical, and mechanical method, it's important to realize that all of these methods will be different with regards to their effectiveness or even their application based on what microbes we're actually targeting. So how many of the microbes are there? What's the nature of the microbes? Do we have high resistance or low resistance? What is the temperature and the acidity of the surrounding environment? How much of that chemical or what type of filter do we require? So which particular mechanism should we choose? And also, the concern is whether or not there could be solvents, organic matter, or inhibitors in the surrounding environment as well, because that can modify how the strategy actually works. Also, specifically, does the particular mechanism require endospores to be destroyed? Can the item, if we're treating a surface, can it be reused? Or a plastic container, for example, can it be recycled? Can it withstand heat, changes in heat, or will it be warped as a result? Or increased pressure, radiation, chemicals? Will it penetrate to the necessary extent? So again, going back to that spectrum of resistance. And lastly, is it cost and labor efficient and safe? So this will hold true for both this chapter and chapter 12. So I would definitely remember this stuff. What are the actual cellular targets of physical and chemical methods, along with even antimicrobial drugs? And they are the following. So you should just think of all of the components that, the main components, especially the essential ones that comprise any given pathogen. So that would be, for the majority of bacteria, they have a cell wall. The cell membrane, all cells will have a cell membrane. They will have a metabolome or a proteome, so they have a ton of different proteins. They also have nucleic acid, so DNA, RNA, and they have ribosomes that make proteins. So for the cell wall, many physical and chemical agents will target the cell wall in such a way that it will break down and the cell becomes weakened. It will become fragile and burst. Detergents will actually poke holes through the cell wall, and also alcohol. Same with the cell membrane, so surfactants work in this way. Now, there are many different agents here that will specifically target a protein, so it will cause the protein to be misfolded. Another word for this is denatured, and then it won't work effectively. Or these agents will target the machinery that is required to synthesize nucleic acid or the protein. So the ribosome, maybe this chemical, or even a lot of antimicrobial drugs target the ribosome. So it could prevent replication of DNA or RNA, or DNA to RNA, which would be transcription, or RNA to protein, which would be translation. And if we're targeting the ribosome itself, it could stop the formation of the peptide bond. And there are a ton of drugs that work this way: tetracyclines, macrolides, erythromycin, chloramphenicol. Also UV radiation and formaldehyde all will target the synthesis of these molecules. So this is just a picture showing you, in the case of a surfactant, poking holes through the membrane and causing the cell to become fragile and, as a result, lyse. Now, heat and also pH changes and heavy metals can cause denaturation, for example, of a protein. So this is the properly folded version. This is a cartoon image of a properly folded protein here that has the appropriate active site for the substrate to bind, okay? And when you expose it to extremes in heat, it causes the unfolding of this protein, and now you don't have this unique active site here where the substrate can bind, so the enzyme is no longer functional. Or it could cause the protein shape to change a little bit, so the active site is unavailable. Or in the case of a heavy metal, which would act as a competitive inhibitor, it will actually bind to the active site itself. So the active site can no longer accept the substrate, and the enzyme is therefore inactive. So now I'm going to go over all of the methods of physical control. And they are changes in heat, so an increase in temperature, a decrease in temperature, which is microstatic. It is not microcidal. Also desiccation, which will dry out the cell, and similar to in the case of changes in osmotic pressure. Or for those of you who are in lab with me, when we conducted the Osmosis Principles lab, you know that we tested the different concentrations of solutes in the agar, and so the higher the solute concentration, the higher that osmotic pressure, and the more likely that cell will be inhibited from it drying out, or this process called desiccation. Also radiation, which will cause changes and mutations in essential macromolecules, DNA, protein. Also filtration, where you're mechanically removing the microbes itself. So as far as heat, moist heat is more effective. So water is a better conductor of heat than air. And in the case of moist heat, it's lower temperatures and shorter exposure time that is required. And the mechanism, the target mechanism here is the protein. So it will cause coagulation and denaturation or misfolding of the proteins. Now we still have proteins that are targeted in dry heat, although this requires higher temperature, so moderate to high temperatures, and it also will result in dehydration. And this is just comparatively here in the case of moist heat, we could carry this out using a machine called an autoclave versus dry heat, lower temperatures, and shorter periods of time. Now some of the terms here which can be applied to a process when you're utilizing an autoclave, or you're applying different cycles of heat to sterilize a particular substance, you would track both the TDT and the TDP over time. So again, usually endospores are the most resistant. So a lot of times we look to see what the TDT for targeting these highly resistant forms would be. So this would be the shortest length of time required to kill all test microbes. And this would be at a specific temperature, whereas TDP is the lowest temperature required to kill all microbes in a specific time designation, 10 minutes. And this is the machine I mentioned before, this autoclave. So it will apply very high pressure, high temperature, and it is steam. So it's moist heat under pressure, and this allows for us to attain sterilization of any heat non-sensitive materials that we would place in this machine. So the steam must reach the surface of the item which we would like to sterilize. The item must not be heat or moisture sensitive, and the way in which it attains this sterilization is to target the proteins, destroy the membranes, and also target the DNA. Now there's a modified form of this where it's non-pressurized steam, it's still moist heat, And in this case, it would be tyndallization. So tyndallization is intermittent sterilization for substances that cannot withstand autoclaving. So here, usually it's a repeated cycle for 3 days of steam for 30 minutes to an hour, and then followed by a cycle of incubation and then re-tyndallization. Now, sometimes this is used for canned foods, lab media, and can be considered a form of disinfection. Also, boiling water is effective at destroying the non-spore forming pathogens. So this would be reaching 100 degrees Celsius for 30 minutes, and it is a form of disinfection. So you're not actually sterilizing whatever. Your boiling will not get rid of endospores. It's just a form of disinfection. So again, it will kill the majority of vegetative bacteria, but not those highly resistant forms. Now, pasteurization, similar to boiling, you're applying heat. There are two different methods here. So there's the batch and the flash method. So this is heat applied to kill potential agents of infection and spoilage without compromising the food flavor or value. So batch method, lower temperatures for longer duration versus the flash. And similar to boiling, this is not a form of sterilization. So it will solely kill the non-spore forming pathogens, lowers the overall microbe count, but will not target endospores or many non-pathogenic microbes. Now, dry heat, as we covered earlier, is not as effective as moist heat, and incineration is a method of using dry heat. So this would be utilizing a flame or electric heating coil, and through incineration you will reduce the number of microbes and other substances. Also, dry ovens can utilize this concept of dry heat, 150 to 180 degrees Celsius, which will cause the denaturation and coagulation of proteins. Now, when you have a decrease in the temperature, this is, as I mentioned before, it's not microcidal, but it's microstatic. So it will inhibit the growth or slow the growth of microbes, but it does not kill them. So refrigeration works in this way, and also freezing, and this can be effective for preserving food, media, and cultures to a certain extent, but it is not microcidal. So you're better off with heat than the cold. Also, desiccation or drying. This is another method, a physical method of antimicrobial control. Now, this works through changes in osmotic pressure. So when you have water that is being removed from cells in a hypertonic environment, this causes metabolic inhibition. And again, it's microstatic. So if you were to place these cells and replenish them from this water poor environment into a water-rich environment, they would start to grow again. But lyophilization is a method utilizing this principle of desiccation where you freeze dry cultures or specific substances, and then again, when you replace them or replenish them with a moist environment, they will start to grow again. But this can be used as a form of preservation. Now, ionizing and non-ionizing radiations are effective forms of antimicrobial control. In the case of ionizing, an example is a gamma or an X-ray, and this can be applied to medical supplies, food products, and basically the way in which it works is it causes DNA double-stranded breaks here. So it has this deep penetrating power that will cause electrons to leave their orbit, causing these DNA double-stranded breaks, which are the most serious form of DNA mutations. And if you have multiple of these, it's very difficult for the cell to restitch these back together appropriately. Using ionizing radiation here to preserve food, you see on the left-hand side it's effective at preventing the growth of pathogenic mold that will spoil the food, as you see on the right. Now, non-ionizing radiation, an example would be UV light. So this is actually the mechanism by which humans can develop melanoma or skin cancer when you're exposed to too much UV light. And when microbes are exposed to UV light, the same particular effect occurs here, where this ultraviolet light will create these mutations in DNA, not DNA double-stranded breaks, but a mutation called a pyrimidine dimer. So pyrimidine is referring to 2 out of the 4 of the smaller nitrogenous bases in DNA. So in DNA, you have a stretch of nitrogenous bases, A's, T's, C's, and G's. The T's and the C's are the pyrimidines. The A's and the G's are the purines. So a pyrimidine dimer is on the same DNA strand. You have a covalent bond that forms between 2 pyrimidines. So it would be between 2 T's or 2 C's. And you see here, this is an abnormal bonding pattern that can be effective at stalling DNA replication and essentially inhibiting and killing the cell. Also, in the case of non-ionizing radiation, this can be used for sterilizing air, water, or other surfaces as well. And here are some examples. So for non-ionizing, mostly for disinfection of air, liquids, commercial use for cleaning rooms, also food processing areas. And this can work well on water, milk, drugs, and vaccines. Also solid materials, foods, tissue, grafts, drugs, and instruments, whereas ionizing radiation works well to sterilize drugs and vaccines, but also sterilization of foods, so including fruits. So we didn't see this down here. Lettuce and spinach, grains, spices, meats, and packaged meals. And also cold sterilization of packaged products such as medical supplies and instruments that cannot be heat sterilized can undergo ionizing radiation. Now, one of the mechanical methods, or the mechanical method of antimicrobial control, is filtration. So this is basically using a filter that has a unique pore size, and as long as the pore size is smaller than the cells that you are filtering out, the liquid will be filtered through this particular membranous filter, leaving the microbes trapped on the other side. So you're physically removing the microbes by passing, and this can be a liquid or gas in the case of a HEPA filter. And this can be used to sterilize both heat-sensitive liquids and also air in hospital isolation units and industrial clean rooms. So now we're going to move on to the chemical agents in microbial control. So these would be termed disinfectants, antiseptics, sterilants, degermers, and preservatives. And all of these that I'm going to go over have the following desirable qualities. So they're pretty effective at low concentrations. They're soluble in water, alcohol, and should be stable. They have broad spectrum, low toxicity, and they're effective. They have deep penetrating action. They're non-corrosive, non-staining, affordable, and readily available. So these are all the desirable qualities. A lot of the chemicals fulfill the majority of these. Now, similar to the physical, mechanical methods of antimicrobial control, we saw that there was a spectrum of resistance, right? So I went over the highest, intermediate, and lowest resistant level microbes. There's also, in the case of chemical forms, high, intermediate, and low level germicides. So just apply the same grouping of microbes to each grouping of germicide. So if we're calling it a high-level germicide, this is one that will destroy the hardiest, most resistant forms of life, which are the endospores. If we're talking low-level, they would target vegetative bacteria, right? And remember, the intermediate had some highly resistant strains of bacteria. also fungal spores and viruses, and even the protozoal cysts as well. So how the chemical agents will work depends on the nature of what is being treated, the degree of contamination, the time that's required, the strength and chemical action of the germicide as well. So I'm going to go over each group here. I believe there are 11 groups. So the halogens, phenols, chlorhexidine, alcohol, H2O2, hydrogen peroxide, aldehyde, gases, detergents and soaps, heavy metals, dyes, and finally acids and alkalis. So I would remember generally each group, and I would be able to give an example of each, know the mechanism of action, so how they work, and give me the application. So when would you actually use them? So first, halogen. So this would include chlorine and iodine. So bleach, for example, will target proteins by breaking disulfide bonds between thiol groups. It's intermediate level, so this would be the target, and it's used to treat water, also wastewater, inanimate objects, but it is unstable in sunlight and can be inactivated by organic matter. Another halogen is iodine. This can be used as an antiseptic. It is also intermediate level and again interferes with the disulfide bonds of proteins. So these are the general applications of the halogen. So it can be used, as you see on the right here in the case of iodine, as an antiseptic generally, and then left-hand side, water treatment for chlorine or surfaces for disinfection. Phenols, they will target cell walls and proteins, and they are low to intermediate level mechanism. Lysol and triclosan are examples. So triclosan is actually now recently been added to soaps as an antibacterial agent. We also have chlorhexidines. This is low to intermediate level, and an example of this would be Hibiclin. So for the hybrid group, we worked with this in our disinfectants lab, and this is used as a skin degerming agent for preoperative scrubs, skin cleaning, and burns. And again, it's targeting the proteins and also will poke holes through cell membranes. So the applications for phenols and chlorhexidine, Hibiclins or Hibitane are examples. So used for preparing skin sites for surgical incisions, also as a wound degermer. So going back to Joseph Lister, right, the idea of antiseptics. And also in the case of phenol, so Lysol, triclosan can be used in a hospital environment, Lysol spray as well. Hexachlorophene can be used additionally to control outbreaks of skin infections. So all those are applications of those two groups. And then we have alcohol. So usually 50 to 95% of ethyl isopropyl solutions, and they're intermediate level, and they will target the membrane lipids and proteins of vegetative bacterial cells and fungi. Hydrogen peroxide, these are used as, or it's used as an antiseptic at low concentrations. And if you increase the concentration, hydrogen peroxide can actually kill spores. So the mechanism of action here, it will produce highly reactive hydroxyl-free radicals that will target protein and DNA. So, and it is toxic to anaerobes. Then applications here, in the case of alcohol, a lot of antiseptic applications for hydrogen peroxide as well. But again, you increase that concentration, it can be used as a type of a sterilant to clean rooms, isolators, space vehicles, even ozone as a form of peroxide for disinfecting air, water, air conditioners, and cooling towers as well. And then we have the aldehydes. So These work by alkylating protein and DNA. So glutaraldehyde and formaldehyde are examples. Glutaraldehyde, low concentration, very effective, and it's a high-level germicide. Formaldehyde as well, intermediate to high, usually in 37% solution, and it is termed a disinfectant, also a preservative, but its toxicity limits its use. For gases and aerosols, ethylene oxide and propylene oxide are examples. They are high-level germicides or chemical agents, and they can be used to sterilize and disinfect plastics and prepackaged devices, along with foods as well. The applications here for aldehydes. So we have a disinfectant for surgical instruments. Also, it is used as a preservative, so it's one active ingredient in embalming fluid. And glutaraldehyde, for example, is used to sterilize respiratory therapy equipment, other medical equipment, dental instruments even. In the case of sterilant gases, sterilization of foods such as nuts, powders, starches, and spices, and even as an air sterilant as well. And then we have the detergents and soaps, so the quaternary ammonia compounds. They're very low level, but they will act as surfactants to alter membrane permeability of bacteria and fungi. And soaps work well to reduce the number of microbes. So again, using a soap to wash your hands would be a form of degerming, and adding a germicide to the soap would drastically reduce the number of microbes. So applications here, the quaternary ammonium compounds can be mixed with cleaning agents to use as a form of disinfection or sanitization of surfaces, restrooms, sanitizing restaurant utensils or food processing equipment, and soaps. This is used for degerming of skin, also routine hand washing by medical and dental personnel, and even in surgical and preoperative hand scrubbing. Again, this is just reducing the number of microbes. And then we have the heavy metals, which we can say have oligodynamic action, so they're very effective at low concentrations, and a lot of them work by acting as competitive inhibitors, so they may block the specific active site of proteins, inhibiting their activity. And examples are silver nitrate or methylate. And as we see here, there's also metallic silver, colloidal silver, even organic mercury. And organic mercury can serve as a weak antiseptic and infective preventative, and also may be used as a preservative in a cosmetic or an ophthalmic solution. Silver nitrate can be used primarily as a topical germicide for mouth ulcers and occasionally root canals. The metallic and colloidal silver rinses for the mouth, nose, eyes, and vagina, and metallic silver to prevent UTIs and even treating toilet seats or medical devices.